Welcome to this video on Nobel Prize for year 2025 in Physiology or Medicine on peripheral immune tolerance. Please watch it completely and like it, share it and subscribe it. The Nobel Prize for the year 2025 in Physiology or Medicine has been awarded to Shimon Sakaguchi, Mary Brunkow and Fred Ramsdell for their work on peripheral tolerance. In this video, I will explain peripheral immune response and go over the contribution of the scientist with respect to the discovery of T regulatory cells, their markers, and the mechanism by which T regulatory cells suppress autoreactive T cells in peripheral tissues, and finally, the significance of their work. So, what is peripheral immune tolerance? We are exposed to a wide variety of pathogens and harmful antigens. Our body releases T cells which protect us by selectively destroying the invading microbes. These T cells mature in the thymus to acquire a selective mechanism to neutralize these pathogens. However, some T cells during their development also show reaction against body's own proteins known as self or autoreactivity. These T cells are also known as autoreactive T cells, therefore. Approximately 60 to 70% of these autoreactive T cells are selectively deleted in the thymus. The remaining autoreactive T cells are either rendered inactive through energy, undergo apoptosis, or are functionally suppressed. However, a subset of these autoreactive T cells develops into regulatory T cells, which we call Treg, by the expression of FOXP3 gene. These Treg cell or T regulatory cells protect us from developing autoimmune disease when released into the peripheral system, and this is known as peripheral immune tolerance. This is also relevant in various other organs like lungs, skin, and GI tract, where these cells rendered hyporesponsiveness of these organs toward the microbe or normal microflora. Shimon conducted a series of elegant experiments to discover T regulatory cell or Treg cells. In experiment number one, when he removed thymus from 3-day-old mice, he found that their organs were damaged and multiple organs were damaged due to autoimmune disease. In experiment number two, when he injected these mice with T cells from healthy mouse, he found that the mice were protected from the damage, autoimmune damage. He concluded that probably T cells are protective against autoimmune disease. In experiment number three, when he deleted selectively CD25 marker from these T cells before injecting into these mice, he found that the protection was gone. Mice were sick again. They developed organ damage. He concluded that probably CD25 cells are protective. In experiment number 4, or the last experiment, he injected mice with CD25 positive T cells, and he found that the mice were protected. They were healthy and there was no organ damage. Based on this, he concluded that Treg cells are CD25 positive. Later, Shimon confirmed that these cells are regulated by FOXP3 gene and they express IL-2 receptor and CD25 abundantly. Therefore, all these three can be considered as markers of the Treg cells. Regarding Brunkow and Ramsdell contribution, they conducted pioneer work on the scurfy mice, which is an autoimmune disease, and human IPEX, which is also an autoimmune disease, which both of which affect males more severely. They identify mutations in both these conditions in a gene called FOXP3 gene. They also map this FOXP3 gene on the X chromosome, explaining why male is affected more severely. In, in addition, they also showed that FOXP3 is the main regulator of Treg cells or T regulatory cells. So, in conclusion, while Shimon Sakaguchi laid the foundational groundwork by discovering T regulatory cells, Mary Brunkow and Fred Ramsdell later elucidated the critical role of FOXP3 gene in regulating Treg cell maturation and function, and its link to autoimmune diseases. Regarding the mechanism of peripheral immune tolerance, when autoreactive T cells are presented with autoantigen, they start synthesizing interleukin 2, which is pro-inflammatory, and chemokines. They attract the T regulatory cells to the site, and they are induced to produce large amount of mediators which are anti-inflammatory, such as interleukin 10, TGF beta, and CTLA4. These these mediators then can silence or inhibit or induce apoptosis of the autoreactive T cells, thereby preventing development of autoimmune diseases. And this is how we develop immune tolerance or peripheral immune tolerance. Regarding the significance, these novel findings provide fundamental insight into the mechanism of autoimmune diseases and cancer. Autoimmune diseases are associated with the suppression of T regulatory cells. Therefore, increasing the number of T regulatory cells can be an effective treatment of these conditions. On the other hand, tumors can manipulate T regulatory cells to suppress immune response and to promote their growth. Therefore, suppressing T regulatory cell can be a promising cancer therapy. So thank you very much for watching. Please like it, share it and subscribe it. Until next time, you take care. Bye bye.